A research breakthrough at the U of A may signal a change in the way we fight malaria and other mosquito-borne illnesses. And the man at the center of that breakthrough, Dr. Michael Riehle, showed U of A President Robert Shelton a little of the day-to-day work that makes these types of discoveries possible.
Well, I know this particular part of your work is focused on mosquitoes and the role they play in transmitting malaria.
Right.
We all know how critical that is. Can you talk a little bit about your approach to this? You're not trying to get rid of all the mosquitoes in the world, I understand, but it's a more subtle, more sophisticated approach.
Yeah, what we're trying to actually do is to replace the mosquitoes that are capable of vectoring malaria or other mosquito-borne diseases with ones that are actually resistant to it. So they're the same mosquitoes with just slight genetic modifications, and they're unable to harbor the malaria parasite. So they would mate with the wild mosquitoes, and the anti-malaria trait would actually be inherited from the ones that we release to the wild mosquitoes. And over time, if we give them a few genetic tricks, it'll actually spread through the entire population.
Oh, okay. Well, I understand we can go and take a look inside some of these controlled areas
well,
to meet and greet some of the mosquitoes.
Yes, let's head into the insectary, we'll look at the mosquitoes.
Okay, well, thanks for bringing me to this sort of inner sanctum of research. I see a lot of plastic containers with many mosquitoes in each one.
Yep.
Different labels. Can you talk to me about what the labels mean? Are they different varieties, different species, different strains, and lots of dates on them?
Right. Yeah, so as I mentioned, we keep two different types of mosquitoes here, one that vectors malaria and one that vectors dengue. And we've genetically engineered a bunch of different mosquito lines, so we have to keep all of those separate. So for the malaria mosquito, we have about 7 or 8 unique transgenic lines, each of which we maintain individually. And so this is actually one of those mosquito lines. This is the Anopheles mosquito.
Yep, all very well known. You can certainly see them in there.
And we actually add a fluorescent marker into their eyes. So if we look at these mosquitoes under ultraviolet light, we can tell one from another. So we can make blue eyes, red eyes, green eyes.
Wow.
And we can actually even mate them. And if we are interested in mosquitoes that have both red and blue eyes, for example, we can look at that.
What is the lifespan of a mosquito? I mean, most of us have no idea.
So it's really interesting. That's one of the projects we're working on. Most of the mosquitoes that are in the field only live about 2 to 3 weeks as adults.
2 to 3 weeks.
And malaria and dengue both have to be in the mosquito developing for at least 14 days, 2 weeks. So it's really just these oldest mosquitoes that are transmitting most of the disease. And so that's one of the targets we're going for, to see if we can reduce the mosquito's lifespan, but still be genetically fit in order to block disease transmission.
Well, let's come on in here now. We've had some time in the insectary. We've seen multiple species, varieties of insects, and saw how they grow from larva to pupa to adult. And now in here we have a series of high-powered microscopes. So tell us how they're utilized in your research.
So a lot of work gets done in this room. This is where we're separating out all of those genetically engineered mosquitoes from the non-genetically engineered mosquitoes. So we have some microscopes here that are attached to UV lights, and we shine the light on them and we can see the red eyes or the green eyes, and separate those from the ones that don't have any eye fluorescence.
And it's physically separating them then?
Yeah. That's actually
one mosquito at a time?
One mosquito at a time, yep.
Wow, that's a lot of labor. No wonder you have so many great students working for you.
Exactly, and this is also where we genetically engineer the lines. So we have micromanipulators that we can actually inject DNA into mosquito eggs at very high magnification. And then we rear those to adulthood, and hopefully a very small percentage of them are genetically engineered.
Well, this is very educational. I really applaud not only the quality of what you're doing, but of course the relevance of what you're doing to all humankind. So thanks for your time.
All right, thanks for visiting.
You bet.